This is a vial of insulin. Diabetics need to inject this insulin daily because the cells in their pancreas cannot produce it anymore. However, what if scientists could grow these insulin-producing cells in a lab and replace the damaged cells in the pancreas with these new, artificial ones? Welcome to the world of stem cells. Now, take a look at this. This is you 40 weeks before you were born, a single cell, what scientists call a zygote. This is also a stem cell. In its most basic definition, a stem cell is a cell that can make copies of itself, what scientists call self-renewal, and specialize into other cells in a process known as differentiation. And so this zygote, in being able to make copies of itself to form the earliest stages of the embryo and differentiate into any cell in the body, is a perfect example of a pluripotent stem cell. You can think of a pluripotent stem cell a bit like this tree here. The cell has a lot of potential. It can become any cell in the body, just like this tree, once cut, could be used as firewood, or used to build a house, or whittled into a sculpture. Depending on what it's needed for, just like a pluripotent stem cell, the tree can become almost anything. However, these pluripotent stem cells can only truly exist in the blastocyst phase of embryos. By the time you're born, the only stem cells you have in your body are what we call multipotent. They can only differentiate into specific tissues, and are typically only used for growth and regeneration, similar to how once a tree is cut down and the lumber milled, the way it is cut limits how a tree can be used. Now, multipotent cells are used for treatments such as bone marrow transplants to treat certain cancers. However, they are much more limited in scope and harder to grow and maintain. Therefore, researchers prefer to work with pluripotent stem cells. The only issue is extracting these from embryos kills the embryo, making them also impractical and controversial. However, what if scientists could reprogram the DNA of a fully differentiated cell and make it go back in time in its development, and become a pluripotent stem cell? Recently, researchers have found ways to do exactly this. The most common method, discovered in 2006 by Shinya Yamanaka, is to insert genes that encode these 4 transcription factors into differentiated cells by way of a retrovirus. These transcription factors deactivate unnecessary genes, causing the cell to act as if in an embryonic environment, simulating a pluripotent stem cell. Now, there are many benefits to these induced pluripotent stem cells, as they're called. If someone needs new tissue or an organ, growing the tissue from cells in their body can reduce rejection rates and make these transplants more accessible. And animal cells can even be replicated to grow meat in a lab. Given livestock alone contributes to nearly 15% of global greenhouse gas emissions, alternatives to meat are becoming ever the more needed in society. Besides being used to grow organs and tissues, stem cells can even be used to model cancerous growth and the progression of other diseases, helping us uncover treatments at a much faster pace. All that said, iPSCs are still a relatively new technology, and still have many flaws. Regardless, given their potential, for our own good, we can only hope some of you will go into stem cell research and one day make a breakthrough.